Hi there, I am Vasireddy, doing my PhD at the BakerIDI Heart and Diabetes Institute. I'll be taking you through the steps that are involved in 3D illustration of histone modification that mark DNA damage and chromatin condensation. In a eukaryotic cell, DNA is wrapped around nucleosomes. Each nucleosome is made up of 4 different types of histones: H2A, H2B, H3 and H4. Gamma radiation induces the formation of DNA double-strand breaks, and this is marked by phosphorylation of H2AX. This phosphorylation of H2AX occurs mainly in euchromatin, that is marked by methylation of H3K4. Now we will be looking at the 3D illustration of these histone modifications in a cell immunolabeled with the phosphorylated H2AX and the methylated H3K4. Let's get started. We begin by placing electrodes of the cell suspension into 15 ml Falcon tubes. Cells are washed twice with PBS by centrifugation at 150 G and resuspended in fresh media. To see the immediate effect of radiation on cellular events, keep cells on ice for 5 to 10 minutes prior to and during irradiation. Expose cells to either gamma radiation or X-rays. After irradiation, dispense 3 ml of the cell suspension into each well of a 6-well plate. For peak gamma H2AX, incubate cells between 30 minutes to 1 hour at 37 degrees centigrade. Use a cytospin machine to place the non-adherent cells onto glass slides. When assembling the cytospin clip, make sure that the hole in filter card coincides with the